Hey, good morning. We're going to get going. So today I have the pleasure of introducing Andrew or Andy Moore. So Andy did his undergraduate degree at Howard College. He went on to University of Pennsylvania with Erica Holzbauer to complete his PhD. And he's been at Janelia since 2018. Initially, when I met him, he was a scientific computing associate in scientific computing. And now he's a postdoctoral researcher in Jennifer Lipping-Schwarz's lab. Along the way, Andy has also been gaining a great reputation in terms of imaging and in terms of really beautiful pictures. He's won a number of, I think, Nikon Small World awards and has had his work featured at the Marine Biological Laboratory several times. And so we're really lucky to have Andy here with us today. And I guess he's going to talk about the FilaBuster. So Andy, please take it away.
All right. Thank you so much for the introduction. And thank you for the invitation, both to you and to Brian. Yeah, I'm really excited. This is my first time actually publicly talking about this work. So this should be a lot of fun. I'm going to be discussing our efforts to generate a tool that allows us to specifically disassemble intermediate filaments in live cells with light. And we called it FilaBuster. So to just step back for a moment, intermediate filaments are one of the three main cytoskeletal elements found in cells. You'll often hear them referred to as the third cytoskeletal element found in cells, in deference to actin filaments and microtubules, which I think are often perceived to be more important, but hopefully I'll disabuse you of that notion by the end of this. This image to the right is a STED image of vimentin in a U2OS cell. What I hope you can appreciate from it is just how bendy and highly curved these filaments are within the cytoplasm. Now, the filaments themselves are actually composed of these fibrous IF protein subunits that are encoded by a fairly large family of genes, over 70 genes in humans. Most of these are the keratins, but you also have cell type specific expression of different IF subtypes like vimentin, GFAP, in mesenchymal and glial cells. You have your axon specific intermediate filaments, your neurofilaments in the brain, as well as lamins, which are a nuclear specific intermediate filament, as opposed to the others, which are all in the cytoplasm. Now, these various IF proteins have at times quite distinct sequences, but they all share certain structural commonalities, namely this alpha helical rod domain in the center of the protein, flanked at either end by variable length unstructured head and tail domains. Now, IF proteins assemble in a relatively unique way compared to other cytoskeletal structures. This process is nucleotide independent, so they don't hydrolyze ATP or GTP like actin or microtubules do. They can actually spontaneously assemble under the appropriate salt conditions, and it's largely regulated by post-translational modification. But the first couple steps are really key. You have an IF protein that then dimerizes with either itself or it heteropolymerizes with a related protein, but you get this polarized dimer that then immediately forms this staggered anti-parallel tetramer, which you can see on the bottom here. What you'll notice now is that any polarity that existed in the IF protein has now disappeared. And consistent with that, IF proteins are apolar. So there are no molecular motors that can move directionally along them from a plus end to a minus end, though the filaments themselves can be cargoes of molecular motors. So here I just have some images showing some of the different morphological features of different IF networks. We have a keratin 13 network on the top left, vimentin on the top right in a COF7 cell. We have the muscle-specific IF desmin on the bottom left, and then you can see an example of a nuclear lamin network, which if you look at some of Mark's work, you can see these actually form really, really beautiful diffraction-limited networks, but they sort of just look like a nucleus if you're imaging them here, like I am with the ARIScan. But in general, intermediate filaments form these really elegant, visually striking networks that are highly cell type specific. So depending on the specific intermediate filament proteins that are expressed, you'll have slightly different appearing IF networks. So they're not just pretty. IFs carry out a number of critically important cell biological functions. They maintain the shape and stiffness of the nucleus. So this is both those lamins inside the nucleus, but also cytoplasmic intermediate filaments can associate with the nucleus via link complex proteins. They're critical for the maintenance of the mechanical integrity of cells, especially the cytoplasm, as well as tissues where they interact with desmosomes and hemidesmosomes to keep tissues intact. And I think one emerging concept that I'm particularly interested in is intermediate filaments actually associate with and anchor organelles. This is true for organelles like the endoplasmic reticulum, lipid droplets, and even mitochondria. And this is a STED image I acquired on the right showing immunofluorescence of TOM20 labeled mitochondria in blue, and vimentin intermediate filaments are labeled in yellow. And hopefully you can see the extent to which these filaments enwrap, crisscross the mitochondria, anchoring them in place. Unsurprisingly, given their wide expression and their important biological functions, IFs are really important for human health, and dysfunction in intermediate filament genes have been linked to several human diseases. So specifically, there are some genetic mutations that lead to skin blistering disorders, cardiomyopathy, but even outside of direct IF mutations, we're finding that lots of different diseases like cancers or traumatic brain injury are characterized by defects in the organization of intermediate filaments like neurofilament, vimentin, or GFAP. But as I sort of alluded to at the beginning, IFs are definitely overshadowed by their cytoskeletal siblings. So here we have an image of a microtubule network on the left, actin filaments in the center, and vimentin IFs on the right. And I could never really figure out why this was the case. Why are they so much less studied? Why are there so many fewer papers? Is it just that they're less interesting? I mean, maybe, but I don't think that's the case. I'd argue that, in fact, IFs are understudied because there is a huge gulf in the tool distribution. So actin and microtubules have tons of highly specific fast-acting tools, whereas intermediate filaments simply don't. So researchers haven't had the implements with which to study these really important cytoskeletal structures. And the reason for that is nature. The natural world has actually provisioned cytoskeletal biologists with great tools for studying microtubules and actin. So here are some of the more famous drugs you may have heard of. Colchicine, for example, was actually isolated from the autumn crocus. Vincristine from periwinkles. I didn't know what that was until a couple days ago. Taxol has been isolated from the Pacific yew tree. So all of these organisms actually produce these chemicals as poisons, but we've been able to co-opt them, derive semi-synthetic versions as well as fully synthetic versions like nicotazole, and these can be used to manipulate the assembly status of microtubules in seconds to minutes. Actin filaments similarly have this broad repertoire of tools. We're not dealing with plants. For whatever reason, actin filament poisons seem to be either marine sponges like Latrunculin A or mushrooms like phalloidin. That is the Amanita phalloides, the death cap mushroom. So if you eat that and die, you can blame phalloidin for stabilizing your actin networks. It's also the gold standard probe for imaging actin in fixed cells. So we put a fluorophore on there. There is no colchicine or latrunculin for intermediate filaments. There aren't any natural toxins or poisons that specifically target intermediate filaments and drive their disassembly. There are no synthetic ones that work particularly well. So people have advertised them, but truly there's no drug you can stick on a cell and see your IFs fall apart like you can with actin filaments and microtubules, which really complicates interpretation of what these things are actually doing in cells. So what are IF biologists left with in this case? So in the absence of good tools, I'd say we have kind of middling tools, and we can sort of visualize this in two dimensions. We can look at the y-axis as specificity, where higher values indicate something that targets IF more specifically. We can also see speed on the x-axis, where we have some things that take days to weeks, whereas some interventions work in seconds. The bottom left quadrant is useless. It doesn't stop companies from selling things that fall into this quadrant, but useless for our purposes. The top left is really where IF biologists, I think, live. These are the predominant techniques. And I think maybe historically more so than any other, RNAi has been used, where you treat cells with siRNA against your intermediate filament protein of choice. Here's an image on the top. We can see vimentin in black, nuclei in blue. And this is a cell treated or plate treated with non-targeting control siRNA. On the bottom, 96 hours with vimentin siRNA. And you can see the filaments are mostly gone, not entirely. You still have squiggles, which is actually the technical term. Those are immature filament squiggles. But something's slightly off about this. This isn't really a one-to-one comparison with a depolymerizer. You're getting rid of all of the soluble protein in addition to the filamentous protein. And in this case, even the mRNA, which may have distinct functions. So in addition to RNAi, more recently, CRISPR knockouts, as well as other knockout approaches have been used. There's been some work using inducible degradation, as well as expression of dominant negatives. But in general, these strategies are somewhat inconsistent and they take a long period of time to affect disassembly of the cytoskeleton. And in that long period of time, you can have compensatory changes that mask the actual phenotype of what these IFs are doing. But still, these are what are mostly used. What if we go into the bottom right corner? These are fast but messy. I don't think these are used routinely as IF disassembly strategies. They kind of fit more into the category of, oh, isn't that interesting? IFs disassemble when you do this. And I think the best example of this is hypotonic stress. If you take cells and just dilute the medium to just 10% in water, what you'll see is the very rapid disassembly of filaments. Here's vimentin. As we move forward, we can see the complete solubilization of that IF network. What you'll also notice is giant vacuoles have opened up. Putting cells directly in water is not a tenable solution. They're not happy. Miraculously, it's reversible if you put them back in isotonic solution quickly. But again, the number of phenotypes that would occur as a result of this would mask any effect of IFs. Another sledgehammer approach is strong oxidants. So incubating cells with millimolar concentrations of hydrogen peroxide completely destroys most IF networks, as well as most of everything else, depending on what you're interested in. And maybe the most specific way of doing this is if you use a PP1, PP2A inhibitor, like oxalic acid or colliculin A. I say specific, but really these work because phosphorylation and dephosphorylation is one of the primary ways that intermediate filaments regulate their assembly and disassembly. But it's also one of the primary ways everything in the cell regulates their function. So if you inhibit a broad-acting phosphatase like this, you're again faced with this issue of specificity. We don't actually know what's happening. So that's where we are. There's no latrunculin. We have a fast and specific quadrant that is completely bereft of tools. So we wondered, could we take one of these imperfect tools and force it up into the goal quadrant? We actually originally did this trying to use some of these dominant negatives. The T3 team was kind enough to help me out with an optogenetic tool. We spent a ton of time on it, and it was just sort of an abject failure for probably planning reasons that I could have worked on a little bit better. But given that approach failed, we switched tacks, and we thought, what if we take one of these fast but messy approaches, like strong oxidants, and did something to make that more specific? And this is really the idea behind the FilaBuster approach. So knowing that these IF proteins are redox sensitive, that the filaments fall apart when you treat with harsh oxidants, and that incubating with H2O2 is obviously a no-go. We can't do that to cells. What if we find a way to target the production of these reactive oxygen species directly to the intermediate filaments, ideally in situ, so that we can target individual networks of individual cells as opposed to bathing the entire sample in ROS? And one idea for this would be to use photosensitizers. So broadly speaking, this biologist perspective of what this is, a photosensitizer is essentially a molecule that when you hit it with a photon, or when it absorbs a photon, it ends up generating reactive oxygen species as a result. It undergoes a transition from the excited state to the triplet state through intersystem crossing, whereupon it interacts with molecular oxygen to drive these ROS. And this is like the trained monkey just repeating the line that a chemist and a physicist have given me. But essentially, we are using a fluorophore to do damage instead of just to visualize a structure. Now, there's a whole suite of tools that have used photosensitizers in the past. These techniques are called CALI, chromophore-assisted light inactivation. And the thinking there is if you shine a very bright light at a fluorophore, you can oxidize whatever is associated with it in a very local neighborhood. About 5 to 10 nanometers is sort of the half damage window of these ROS, and inactivate it. So we said, what if we do the same approach, but try to refine it a little bit? So we started by looking at kind of the easiest intermediate filament protein class to look at, which is vimentin. They're very simple, they're well characterized. The complete filament structure was recently published, so it seemed like a good starting place. And we used the halo tag self-labeling system to localize specifically to intermediate filaments. So this is our tool in the top left. This is a fusion between vimentin with a C-terminal halo tag. When we express this in cells, we reason that it would co-assemble with the endogenous vimentin network to form mature filaments that are lined with these halo tags as sort of these little probe attachment sites built into the native network. We then labeled with photosensitizer halo tag ligands. Fortunately, the Lavis lab has generated a number of these. They're bright and they're quite potent. JF570 is sort of the flagship photosensitizer dye that we started with. And then we thought, simply illuminate with light, excite the photosensitizer, generate ROS locally, does that drive disassembly? Can we effectively miniaturize or focus this Ross effect directly onto the filaments? So to begin to answer that, we had to do some validation first. The first big question was, when we express our vimentin halo tag probe, does it even incorporate into the endogenous network? We can't take that for granted. So here's a COS7 cell that's double labeled with our vimentin halo tag probe. And this is vimentin M emerald on the right. This has been knocked in, so we're looking at the endogenous vimentin network there. And we see very close co-localization. So it seems like our tool is going the place where it's supposed to go. We also did follow-up staining where we expressed the vimentin halo tag on the left and also stained for endogenous vimentin. And once again, we're not seeing any kind of aberrations, increased bundling or non-specific association that would suggest that our tool was not integrating into the correct location. But just because something looks good by light microscopy doesn't necessarily mean that it is actually integrating into the filaments in a non-perturbative way. So together with Ohad Medalia's group, we used cryo-electron tomography to visualize the individual filaments with the halo tags incorporated. So here is a tomogram. This is just kind of a stack going through it. We can see those straight lines running are the vimentin intermediate filaments, about 11 nanometers wide. And those little black dots that you can see all around them are actually the halo tags hanging off of the C-terminus surrounding the IF. Maybe a little tricky to see in there. So we have a denoised projection here where the vimentin filament is running through the center, and each of those pseudo-colored red dots is a halo tag hanging off of the filament. We calculated the distance from the halo tag to the center of the filament. We found an average of about 12.27 nanometers, which puts it well within the half damage range of some of these ROS that are going to get produced by our photosensitizer dyes, given that that's measuring to the center as opposed to the edge. And we also found that the halo tags integrated throughout the entire network. They weren't just clumped in one portion. We're getting a pretty good coverage map of halo tag ligands throughout each of the individual filaments. So it seems like the tag is going to the right place. It's integrating. The filaments aren't fraying or falling apart, but does the idea actually work? And yes, I wouldn't be here talking about it if not, but here's a COS7 cell that we labeled with JF570. In the first frame, we can see, this is the first time that we have exposed the cell to light, which is very important. We see a very nice, well-integrated network where the vimentin density is a little bit more pronounced in the perinuclear region. And as you go into the periphery of the cell, it gets a little bit more sparse. As we begin to image, we see within about 8 frames, the complete disintegration or solubilization of this network. So I'll play that a couple times. This isn't photo bleaching. We can do a few different image analysis checks to look at this. So we see that as that non-uniform IF distribution spreads out throughout the entire cell, we see a decrease in the coefficient of variation of the image, measured with the standard deviation divided by the mean. Also importantly, we can actually measure the radial distribution of vimentin mass over time. So in frame 1, shown by that green line, we can see that pronounced perinuclear distribution indicated by that large hump dropping off over time. By frame 11, we can see that we've sort of flattened the curve, to borrow a familiar term. We have this uniform soluble vimentin IF spread. So this was an ARI scan image that I acquired with a 561 nanometer laser line with a COS7 cell, but this isn't only something that works with ARI scan or COS cells. We validated it across different cell types and imaging modalities. Here we have a mouse embryonic fibroblast, which only expresses vimentin. They don't have mixed cytoplasmic IFs. And we can see, again, imaging here. This is now wide field, the complete disintegration of what was previously a filamentous network. And I'll play this movie a couple of times because you can actually see over about 1 minute here the process by which those filaments break down into fragments. And simultaneously, we see an increase in the soluble background signal indicating free vimentin subunits diffusing. And this can also be quantified by looking at the local coherency of those filaments, which drops down precipitously over time. So one thought that we had was, while it appears very much like we're fragmenting the vimentin network, it's possible that we're just losing our tool. Maybe the halo tag is popping out of the filaments and the endogenous network is completely fine. That's a big concern if that's indeed what we're doing. So to evaluate that, we went back to our cell line that has endogenously tagged vimentin, and we co-expressed halo tag once again. We then imaged. For clarity, I'm only showing the endogenously tagged vimentin right now, but we are simultaneously imaging both probes. But when I play this movie, hopefully you can see the intact filaments undergo very clear fragmentation over 7 minutes and 44 seconds, but I think it's only about 15 frames there. I have to double check. Here we can actually see both channels side by side, if interested. That's the probe on top, the JF570, and the endogenous vimentin simultaneously breaking apart. So it seems mission accomplished. We have a baseline FilaBuster implementation where we can use light to trigger disassembly of intermediate filaments. 24 hours later, those cells are still alive. We did biochemistry with them. It doesn't trigger runaway oxidative damage to the cell in the same way that hydrogen peroxide does. But it's kind of a niche implementation. We have to express vimentin halo tag. So you have to be interested in vimentin. And we only use one dye. We wondered, maybe there's a way we could make this a slightly more versatile technique. Taking advantage of how modular this is, could we swap out one of these steps so that maybe this approach could be more useful for an IF researcher interested in something else? And the first thing that we asked was, all right, well, what if you don't want to directly tag your subunit? Oftentimes, structural proteins don't appreciate when you stick a giant fluorophore onto them. Fortunately, this vimentin halo tag was well tolerated, but there are lots of examples of fusion proteins where they don't form correct networks. So we asked, is there a separate mechanism or a parallel mechanism where we can target halo tag to the filaments without actually labeling the structural protein? And we decided to use a nanobody strategy. These are cell expressible camelid nanobodies, and they're quite small, smaller than the halo tag vimentin fusion. We fused the halo tag to the C terminus, expressed it in cells, and found that it beautifully localized to the endogenous filament network. So on the top right here, that's the endogenous filaments labeled with emerald. And on the bottom, this is our vimentin halo tag nanobody labeled with JF570. One thing to note, the signal to noise of the filaments is not as clean. It's not as high as it is with the direct tagged IF subunit. And that's because you're constantly exchanging your bound and your unbound antibody, which is critical. If it was too tightly binding to the IF, it would be interfering with its functions. But despite this lower signal to noise ratio, we found that it still worked extremely well. So as we imaged, we began to see the disappearance of the antibody as the filaments broke apart. And as you can see at the top, the vimentin M emerald signal began to fragment as well. So we have an alternative mechanism besides just labeling the subunit. And fortunately, nanobodies for at least 7 or 8 other intermediate filaments have been described and exist in the literature. So this is hopefully a tenable strategy if you're interested in GFAP or Nestin, for example, to reproduce this approach. Now, we showed JF570 halo tag ligand, which is sort of the flagship photosensitizer dye that Luke's lab designed. But you can imagine a number of circumstances in which you wouldn't want to use the red channel for this. You might want to co-express, say, Tom20M cherry to see the effect of vimentin loss on mitochondria. So we wondered, is it possible to find a spectrally separable photosensitizer, ideally red shifted, so we can use even longer wavelength light, which would be less damaging, could penetrate farther into tissue. And it just so happens that Luke's group had already made a dye like this, and it was just looking for an application. So I was chatting with him about this project, and he was like, yeah, give it a shot. This was originally called JF608 dibromo. It's a variant of JF608 where they added 2 bromine substituents. It's a little bit small to see in the top left there. But what effectively did is it harnessed the heavy atom effect to convert this molecule into a more efficient photosensitizer. It also shifted the excitation maxima from 608 to actually 639 when it was bound to halo tag ligand, which was a perfect match for the 639 nanometer laser that we had on the area scan. And it also happened to be fluorogenic. I'm not sure that helps us so much, but it's really fascinating that it became a fluorogenic molecule, I think. But here we have a meth cell labeled now with JF634, our far red fluorogenic photosensitizer dye. We illuminate with 640 nanometer light here by wide field, and it is just as effective as the JF570. We see a small amount of bleaching, the disassembly of intermediate filaments. It's a little bit clearer in the higher signal to noise area scan movie here. You're in a cough cell, but over subsequent frames, we can see the complete dissolution of these structures. So we have an approach, and it works. But there's a problem. It's so potent that certain limitations are introduced in the experimental design. You cannot look at your cells through the eyepiece with your red epi light without inducing disassembly. So actually, we can see that here. Let me just play this first. So this is another movie of a COS7 cell where the moment you begin imaging, your intermediate filaments disappear. So you have no way of tracking IF dynamics beforehand unless you co-express another conventional IF protein, waste another channel. And once these IFs have been disassembled, as you continue to image, you're generating a lot of reactive oxygen species and potentially damaging the cell, which is not ideal. Another problem is we envision this tool as being capable of spatial control in addition to temporal control. So we wanted to be able to just hit a small portion of the network with light and then image the rest of the IFs around it. And you can definitely hit a small portion of light here and disassemble the IFs. But when you zoom out to the rest of the cell, the moment you begin imaging with 570 or 634, you disassemble all the filaments. So you lose the ability to discriminate your disassembled filaments from your assembled filaments. So we wondered, could we refine this process with maybe better ligands or a better approach to have precise spatiotemporal control? And we reasoned that we were looking for really potent photosensitizer dyes just to make sure that this would work. But maybe we could just use conventional regular dyes and just dial up the laser power a little bit to trigger the same type of reactive oxygen species production that the photosensitizers do. That's where we kind of got this idea of a dual use dye. So in standard imaging mode with, I'd say a low energy dose, but this is sort of a conventional energy dose that you would use for live imaging, a ligand like this would allow you to monitor vimentin dynamics for as long as you'd like. Then in FilaBuster mode, you would raise the energy 2 orders of magnitude, you increase your laser power, and then at the moment of your choosing, you would be able to drive IF disassembly. Maybe this is a pipe dream, but again, Luke is upstairs. He's just made so many dyes that he was happy to just give me a ton of them, and I started screening. So we started with one of my personal favorites, which is JF657. It's a incredibly bright, stable, far red carborrhodamine dye. And here in the top, we have our standard or safe imaging mode. This is a meth that expresses vimentin halo tag that I've labeled with this far red conventional dye. And what you'll see is over these 601 frames, we maintain a bright, high SNR image with absolutely no fragmentation of the intermediate filaments whatsoever. You can image these for 12 hours, higher energy doses than just 23.4 joules per centimeter squared, and they're pretty happy. You don't see any disassembly whatsoever. Now, if we then dialed up the laser power in FilaBuster mode, going from 0.02% laser power to 2.0% laser power. This is 2.7 milliwatts that we measured at the sample plane. We found that within 151 cycles of illumination, this conventional dye that isn't supposed to damage our intermediate filaments could, under the appropriate conditions, drive IF disassembly. So by 151 frames, we begin to see the beginning of the fragmentation, and by 300 frames, we could drive total FilaBuster dissolution of the vimentin IF network. 657 worked, so let's just throw everything at it, which is what I did. Here is a cross-section of some of the dyes that Luke has generated. There's too much information to process here, but the important point to note is these run the spectrum from 488 excitable dyes to far red laser excitable dyes. Those indicated in red are designed as photosensitizers. Those indicated as black are not. They're just bright, stable halo tag ligands. And we tracked the number of cycles of illumination that was required until we saw IF disassembly. Again, a lot of information here, but what I want you to take into account is the higher values indicate dyes that take longer to disassemble. These are dyes that are less efficient filibusters, but also dyes that have bigger separation between the imaging mode, where we can image them forever without breaking up our filaments, and the FilaBuster mode. And the size of that window actually is kind of nice to have, because it gives you confidence that your tool isn't affecting your intermediate filaments when you're using it in safe mode. What I found kind of interesting was at these higher energy powers that we did the screen in, some of the conventional JFX 549, JF 554 were as efficient as some of the photosensitizer dyes as driving disassembly. But we also had some over here at the right, like JF 585 and JFX 650, which were inefficient. So we reasoned these might be really good candidates to bring back for that focal disassembly that we were unable to get with the photosensitizer dyes. Here are just a couple of images, or movies rather, of these conventional JF dyes disassembling the vimentin halo tag network. That's JF 552 on the left. And you'll notice these are higher laser powers, but we're not significantly bleaching these dyes. In the scheme of things, we're not hitting them with so much laser power that the dyes are losing their signal, but it is considerably more than you would want to use for a standard imaging experiment. And this is JF 635 by lattice SIM microscopy on the right. Okay, so now armed with this new repertoire of conventional dyes that we know can trigger disassembly at higher power, we once again went back and said, all right, can we now do this spatially controlled focal disassembly? So we labeled our halo tag ligands and imaged them for long periods of time in safe mode. They form perfectly normal networks. We then focally irradiated a region of interest in the cell using that increased FilaBuster power. And what we found was the complete and contained dissolution of IFs within that site, allowing us to then query what role different populations of IFs have in the cell. So we had originally tried to just make a tool that would let us disassemble IFs, and we ended up with something that allowed us to do it in a spatial and temporally controllable fashion. Here on the bottom is just a sort of a representative montage. The second frame is actually after irradiation, so you can see they're not bleached within the box, but within 1 minute and 15 seconds, the signal drops due to the fragmentation and diffusion of the fragments out. This works exceptionally well in intermediate filament structures that are notoriously challenging to disassemble, even with those fast and not very good approaches. So really, really dense bundles. These bundles are sometimes even inaccessible to antibody staining, even after you treat with, you fix them, you treat with Triton, they're just so dense with so many filaments, it's really hard to get in there. But given that we have baked in our sleeper cell of halo tag ligands into the endogenous network, it's accessible to us. So we can briefly irradiate with 639 nanometer light and cut that vimentin bundle in half. We can cut single filaments in half and see that they retract and pull back as a result. So we can begin to use this to query biomechanical functions of IFs and cells. This gnarly structure right here is a perinuclear vimentin knot or vimentin cap. These are really challenging to get into, but with FilaBuster, we just essentially cut a stripe through the middle and saw as the two halves of the now cleaved knot separate out. And you can see some of that soluble vimentin signal diffusing throughout the entire cell. This is my favorite movie on the right. This is using the vimentin nanobody approach here. I like it because it looks like a little guy, kind of looks like a hangman guy. Shortly into the movie, I eviscerate him. Is that legal? I don't know. I don't know. But what hopefully this jokes aside comparison shows is the tool is robust and it could work for incredibly dense IF populations as well as for single filaments. It is the resolution to eliminate your entire network very quickly or target just one particular structure, say associated with the mitochondrion or ER that you might want to query the effect of. So far we've seen the baseline implementation with the photosensitizer works. We've developed parallel strategies using alternative delivery with nanobody, alternative photosensitizer dyes. We've benchmarked conventional dyes that are compatible with both standard and FilaBuster mode, and we've showed efficacy in this focal disassembly of subsets of the IF network. But I think one question that obviously people want to know anytime there's a new tool is when and where does it fail? I've shown you a lot of success cases, but where does this not work? So there are a few controls that I think just very briefly are worth mentioning. We took great pains to target the halo tag directly to the filament surface, but we never tested. Maybe you can just put cytoplasmic halo tag in your cells, irradiate it, and that ROS is just enough to disassemble your IFs at baseline. I mean, surely H2O2 works. But that's not the case. So here on the left, we have just cytosolic halo tag labeled with JFX549. On the right is endogenously tagged vimentin. We irradiate this with a light dose that's substantially higher than what is necessary to drive vimentin halo tag disassembly. We even photo bleach the whole cell at the end, and we see absolutely no loss of vimentin IFs, despite the cell beginning to look kind of sick and mitochondria beginning to round up. So there's sort of an untenable light dose using this approach. We wondered, is it possible to use the FilaBuster approach on other structures? So chromophore-assisted light inactivation has in the past been used for things like actin and microtubules at very, very high energy doses, much higher than what we're using here, often multiple orders of magnitude higher. But could we use the same targeted specific halo tag approach with actin filaments at reasonable light doses? So on the left here, we have LifeAct halo tag labeled with our potent far red photosensitizer. On the right, we have vimentin M emerald. It's challenging to say, but I cannot envision a world in which anybody would want to use an approach like this over something like latrunculin or any of the readily available actin disassembly approaches. It appears as though we're not getting substantial actin disassembly. There are very clearly some stress fibers that persist at the end of this movie. But I'll play it one more time. If you just look at the mitochondria in the negative that you can kind of see, you'll see they begin to sort of swell and pop as the movie goes on. The cell is going to die not long after this. And the light dose used here was, I think it was like something like 20% laser power on the far red. We have the actual measurements accompanying it, but suffice to say, much higher than you want to use. So No, this isn't something, FilaBuster is not a tool that we envision will be applicable, generally speaking, to other cytoskeletal elements like actin. But I keep talking about halo tag. What's my obsession with halo tag? Why can't we use another mechanism for generating ROS at filaments? And certainly other mechanisms exist. There are a whole suite of genetically encoded photosensitizers, like the killer red proteins or the supernova 2 proteins. So we express vimentin supernova 2. When excited, this should generate ROS, though admittedly at slightly lower levels than the halo tag, and it didn't work at all. So that was supernova 2 on the left, bleaching out at the very beginning of the movie. This was our vimentin M emerald, completely intact by the point the protein's gone. But we thought surely snap tag will work. It's very similar to halo tag. Don't tell any of the halo tag or snap tag people I said that. But conceptually, it's a self-labeling protein. It uses dyes that are quite similar. Though admittedly snap tag has lower labeling efficiency. So we figured this should work, but it didn't. So here's the vimentin snap tag labeled network. We labeled with JFX554, but the sort of the snap tag ligand version instead of the halo tag ligand version. And over an hour of high speed, high power imaging, we got maybe an intermediate phenotype where things started to look a little hairy and maybe there was some fragmentation, but not that dramatic rapid effect. So there's something very unique about the halo tag and how it can drive this potent disassembly that we just simply can't reproduce with other photosensitizer approaches quite yet. So much of this work was done in vimentin because it's easy. I'm familiar with it. Tools exist. But an important question is this just a vimentin FilaBuster or is this truly a filament buster? Maybe this is the first time I've explained that. Filament buster. That's how we got it. So it is generalizable as far as we can tell for all the use cases that we've looked at so far. So we generated fusions between other type 3 IFs, GFAP, which is expressed in astrocytes, upregulated in brain injury, peripherin, a peripherin neuron, intermediate filament, desmin, the muscle-specific IF. We followed the same exact strategy of labeling with a dye and irradiating, and we found that all of these type 3 IFs disassembled with comparable kinetics. Here's a GFAP network fragmenting on the left. Here's another lattice sim image of the GFAP network fragmenting over time into those small remnants. Desmin here on the bottom left, and then peripherin on the bottom right. So there seems to be a common mechanism at play, or at least a common sensitivity that all of these IFs have to ROS that's being exploited with our tool. Now, all of those type 3s are relatively similar. One of the most important tests we had to do is what about a keratin, the largest class of IF proteins that are structurally a little bit different. They're heteropolymers, they have unique properties. Well, using keratin 18, labeled with halo tag, JF552, we irradiated. It took marginally longer, a little bit longer than the type 3s, but we also saw complete loss of intermediate filaments. But curiously, it looked different. We didn't see scission and fragmentation of the filaments like we'd seen. Instead, we see kind of the bubbling up of, we almost tempted to call condensates or these puncta along the intermediate filaments, almost like the keratins were being sucked out of filament state and entered into these dense structures. This actually isn't too different from one of the phenotypes that you see with keratin when you treat it with those phosphatase inhibitors that I described earlier. So we're still screening other IF subtypes. It'd be really interesting to look at neurofilaments, to look at nestin, and some of those other sort of rarer flavors of IFs, but we only have so much time. So given this tool that we developed, I think one lingering question is, what new biology can we actually learn with this? Is this of any value, or is this just sort of an exercise in a biologist trying to make a tool for the first time? And I think legitimately we have learned something so far in cultured cells, but there's a lot that can still be done with these tools, taking them into an in vivo context or taking them into a cell type more specific for the IF. But one of the first things that we found was upon acute VIF fragmentation, we see very rapidly there are cell shape changes. Now, these changes don't immediately precede apoptosis or cell death. They occur even with the gentler dyes that we see upon IF fragmentation. And I should provide some data for this, but the vast majority of cells are still alive 24 hours later. We've done pulse chases to look at vimentin expression. But the general phenotype that we're seeing is a progressive rounding of the cells and retraction of some of these extended processes. Now, I'm tempted to say it's somewhat similar to the mesenchymal to epithelial transition, where you downregulate your vimentin, you see consequent cell shape changes, but it's challenging to say. This is just a sort of a taste of a possible phenotype that could be pursued, but that maybe wouldn't be appreciable with conventional approaches. What was a really striking phenotype that we saw there's thought to be this role for vimentin intermediate filaments in caging organelles and controlling their positioning. Now, here is a movie where we have vimentin halo tag JF634 in the center and just a transmitted light movie on the right. And what you can see on the right next to the nucleus at the bottom is just sort of what would sort of historically called the endoplasm. It's a mass of perinuclear organelles. Who knows what they are? There's some mitochondria, ER, some lipid droplets, some peroxisomes. When I disassemble vimentin, what you'll see is sort of the radial outward motion of all of those organelles. It's almost an advection of organelles from the perinuclear area. So I'll play that now. And this is something that we can quantify quite easily. So here we have traced the mass of the perinuclear organelles. At frame 15, after the vimentin is disassembled, we can see that there has been a pronounced increase in the area encompassed by this. Particle image velocimetry also can be used to quantify the motion of the organelles by transmitted light. But this supports a model where vimentin intermediate filaments function essentially as a cage to maintain the perinuclear distribution of organelles. We looked also specifically at lipid droplets, which IFs have been known to associate with. In this movie, we image in standard imaging mode on the left halfway through, and then we increase the laser power to FilaBuster mode. If you look at the right, those black dots are each lipid droplets. Shortly after we increase the laser power and vimentin disassembles, I hope you can appreciate that there is an outward motion of those lipid droplets. So they lose their vimentin-dependent confinement, which maybe is also obvious in this movie we have that's a binarized position of those lipid droplets over time. Now, only the lipid droplets that co-localize with vimentin undergo that shift in position. Those that were trafficking out in the periphery do not. Again, supporting the idea that loss of IFs is associated with the change in localization. If you tracked one of those lipid droplets over time, as we did here, you could see for the vast majority of the movie, they're essentially confined and stationary, but at the moment of that fragmentation, you can see this highly directional advection. MSD analysis gave alpha values close to 2 for this type of motion and much more confined subdiffusive type motion prior to that. But I think you can see it sort of from this cartoon and just from sort of the ensemble tracks here, where at baseline, we have this vimentin cage surrounding lipid droplets. And after we fragment vimentin, we can see them shoot outward. Previously, you would have to do a knockdown or a knockout of vimentin, wait 72 to 96 hours, and then compare distribution of lipid droplets. And it's entirely possible that over that time period, compensatory mechanisms would have taken place, which would have changed their distribution at baseline. Another stark phenotype that we see is that upon global disassembly of vimentin IFs, we see a structural reorganization of the endoplasmic reticulum. So here we have the ER labeled with snap tag Sec61 in the center. On the right, we have our vimentin halo tag FilaBuster probe. What you can see in this initial frame is this ER is highly tubular in the periphery. It has that very characteristic reticular morphology with those polygonal voids separating the tubules. When we disassemble the vimentin, we see the expansion of planar sheets, which is not dissimilar to a phenotype that was described upon loss or mutation of keratins. That there is a shift from more tubular ER to more sheet-like ER, but we can see that this process occurs nearly instantaneously with loss of IFs. A little teaser for another project I'm working on. We previously found that intermediate filaments can actually confine and trap ER tubules within structures that we call ER tubule matrices. Here's an example of one right here. That's a high density of ER tubules in the center. That's a vimentin knot or entanglement on the right. We can do the same trick. We bleach the vimentin on the right. The vimentin focally disassembles. And that entangled, highly confined tubular structure that was so dense in the first frame essentially reverts back to general standard peripheral tubular ER that's no longer clustered. Okay, this one's good. This is probably the clearest example. Maybe I should have displayed this. That's a matrix in the center or a tubule cluster. On the right, that's vimentin just grabbing onto those ER tubules, clustering them together like glue. Halfway through, I'm going to just cut a stripe through the vimentin. And if you look at the ER, you'll see the cluster completely disappears there. There's the bleach, and now we've effectively cut it in two. So organelles like the ER respond very, very rapidly to this acute IF disassembly, which is something that we really did not know beforehand. So in summary, we have this FilaBuster tool, which is, to our knowledge, the first rapid, but also specific mechanism for disassembling IFs in cells. FilaBuster affords us spatiotemporal control, so we can eviscerate the little man. It's generalizable across IF classes. We showed this with multiple type 3 IFs in addition to cytokeratin. So there's promise that this could be an approach that would work for other IF subtypes as well. And finally, it's modular and highly versatile. So we can use a range of different Janelia fluoride dyes. You can multiplex this with other markers in your experiments. And we're doing some tests now to see if we can carry out these in either organoid models or in vivo moving forward. So this was kind of a whirlwind project that took place over kind of the last 14 months. And really, the two people I think that I have to acknowledge first and foremost is my co-author, Tommy Krug from Harvard. Tommy was a visitor who was coming to the lab for a completely different project. But we stumbled across this effect a couple days in, and we kind of just said, drop everything. This is what we're going to do now. So it's been a delight to work with him. And I think he's on the call. So it's I should definitely say that. Also to my postdoctoral advisor, Jennifer Lippincott-Shorts, who has been incredibly tolerant of this complete redirection of what the research initiative was. We were all focused on this vimentin ER thing, and she was patient enough to let us just say, all right, let's take a year and just do something totally different. To all of the collaborators at Janelia, Alexander, who designed and generated the vimentin M-Emerald knock-in line, to all the folks in Luke's lab, John, Anthony, Sarah, who designed and made available these amazing halotag ligands and snap-tag ligands that I used in the project, which was outstanding. I didn't have an opportunity to go into it, but we've done mass spec to get a little bit more at the mechanism of this with Nan at the mass spec core here, who's instrumental in that. And Dave White's Tommy's advisor. And then, of course, the Medallia lab, both Ohad Medallia, his postdoc Simon, who did the cryo-ET and showed us the structure of those halotags in close proximity to vimentin. And I don't think I've ever seen talk about vimentin without Bob Goldman in the acknowledgements. Bob was Mark's postdoctoral advisor and has been a tremendous sort of mentor and advisor to me in this project. I remember going up to him at ASCB and just saying, teach me vimentin. Why doesn't anyone study this? And he's been very accommodating. And thank you to all the folks at Janelia who assisted. Satya for sharing her immense knowledge of intermediate filaments, as well as Gokul, Michael, and Brian for helping me out with measurements of laser power and how to make sense of calculating energy dose in both confocal and wide field microscopy. More details of which are in the preprint, which I should say was posted on bioRxiv a few weeks ago. So feel free to check that out there. And with that, I'd be happy to take any questions. Thank you.
All right, I had a quick question.
Eddie. That was amazing. I have a question because the mitols are like very close to the vimentin network. Are they affected by the ROS that is also very close?
Yeah, it seems like when done correctly, I should say, the answer is no. In fact, we have, in addition to using dyes like TMRM to look at mitochondrial membrane potential, we also use CellRox Deep Red to measure reactive oxygen species. I think it's superoxide is the sensor, and that actually localizes to mitochondria because it's one of the predominant areas where that's being generated. It's not the best ROS dye on Earth, but in this time lapse, you can see we disassembled vimentin in white on the top, and we didn't even see substantial changes in ROS production in the mitochondria local to them. But I can also say, if you use JF570 or JF634 and you keep imaging your cell, you absolutely will see phototoxic effects on everything. So calculating empirically in your system the energy dose required to get this effect and using safe mode instead of FilaBuster mode when you can is critical.
So,
Oh, go ahead.
okay. All right. I thought it was super interesting that you saw these rapid cell shape changes upon disassembly. We've had some work where we see that keratins can actually organize myosin activation. And I was curious if you think that there's any connection with like RAC1 or ROA, and if you consider going down that road.
Absolutely. So we're just beginning to go down this road, but it is my suspicion that the cell shape changes are not entirely due to loss of filament integrity, but rather due to the sudden and dramatic increase in the soluble pool of vimentin. All of these soluble, they're probably tetrameric, but I can't say for certain, vimentin subunits are released and diffused throughout the cell, and they're actually associating directly with actin. So we can see them, look, I don't actually know the cameras. We can see them directly accumulate on actin filaments, on stress fibers. This is an irreversible process. The filaments don't reform, but about 2 hours later, you can see that stress fibers that are decorated with those fragmented vimentin subunits shear and break apart. So I imagine there's some direct association between vimentin and the actin cytoskeleton that's driving that. It'd be nice to look at like a rack sensor or a row or CDC42 sensor to see what's going on signaling wise there. But vimentin does bind actin, so it could be a direct interaction that's causing. Thanks.
Oh, go ahead. My question was, have you tried using actin instead of Light-Cont?
We haven't yet. That would be a good experiment. It would be more direct. We figured the nanobody working for vimentin was sort of the proof of concept that you could have a binding protein be the delivery mechanism. But yeah, to your point, it may be that Light-Cont has such a quick landing time on and off that you may not have enough of it at a given point. Actin may work, but of course you run into the problem again that I mentioned with tagging a cytoskeletal protein,
Right.
Sticking halo tag on actin. I mean, it's certainly in yeast, sticking an FP like GFP on actin interferes with formin-mediated assembly. It's possible that you might be screwing up your network, even if you endogenously tag with halo tag.
But your data, your image, you still could see stress fibers at the core of it. So probably because Light-Cont is onto the surface, so you kind of disrupt the surface, but not the whole thing. So it kind of works, but just because Light-Cont is on the surface only, and also Light-Cont is a membrane-bound protein, the other part of it. So maybe that's why, because Light-Cont is a short part.
Just the right. The full.
Maybe that's why you also see the morphology change in mitochondria as well, because so membrane protein.
It's possible, though. I will say the morphology change is pretty consistent with, like, if you have your cytoplasmic halo tag and blitz it with light, you see similar types of mitochondrial explosion events. They're usually the canary in the coal mine of phototoxicity in the cell, but it's worth looking into. And certainly I am a believer that actin associates closely with mitochondria. So there's a population there.
The other one is the keratin. Can you try both, type 1 and type 2 together?
No.
Because in general, even if you deplete, if you overexpress just one type into the cell, it will pull out that one, and the other one will just form both sides. It just the pair. Deplete one, shut the other.
Yeah, it's a great question that we should be looking into.
Yeah, the feature look exactly like that. Just
the blobs,
Right.
Yeah. I think one question I'd have from that is your interest in querying the mechanical changes that happen when you lose intact filaments? If so, even if you're doing something artificial, your filaments will not have the same mechanical properties. They will break down. So it may be achievable in the end, but certainly we found when overexpressing keratin 18 in some cell types, it simply just did not form filaments and went directly into condensates. In the cost, it formed beautiful filaments, probably because, I'm sure you know, but I can't remember offhand which keratin it copolymerizes with. But I think in that case, it is just simply the filaments.
And when you disrupt the vimentin, what the actin stress fiber looks like? Do you stain them?
I took the movie out, unfortunately, but what you see is that all of those, what look like dissolved soluble vimentin, they begin to coalesce on actin stress fibers. They decorate them really, really heavily. This actually is a phenotype that's been seen with ULF mutants with vimentin, as well as after osmotic stress. But they envelop their surface, and after about a couple of hours, they just simply shear, and you can see them snapping apart. But we haven't looked at other actin binding proteins to see, or myosins to see if they're also localized there.
Or like the edge, it looks like forming more lamella and lamellipodia. I wonder what are all the R2, and other protein looks like.
Frankly, I think it's binding plectin immediately first. So it goes to focal adhesions, it goes to nuclear envelope, but it goes a little bit to the cortex. But I'm not confident enough to say that without doing staining. But it could be affecting branch out.
We have a question on Zoom. Juan, do you want to unmute?
Yeah. So I had a question, like, let's say I have a different structure that I'm interested in disassembling, maybe that I think might be sensitive to like targeted oxidative stress. Do you think like just seeing how well it responds to peroxide is already a good metric for whether such a strategy will work? Or do you think that you really need to implement this to see whether this targeted oxidative stress is like especially going to work?
Yeah, that's a great question. Honestly, I don't have good intuition to give you an answer. I already knew that these structures were really sensitive to oxidants and electrophilic toxins, so I went ahead and tested it. I don't have a good sense of what the local concentration within 5 nanometers of your photosensitizer is versus what you would need to get on like the millimolar level of bathing your cell in H2O2. But my feeling with lots of experiments is just, yeah, I would just try it. Maybe try it with H2O2 and see what happens. But you know, it's pretty straightforward to implement this with whatever your tool is. You just slap your halo tag and you're doing Cali or chromophore assisted light activation. So give it a shot. Yeah.
Andy, can I ask you, first of all, beautiful. But what I find really cool is when you switch into FilaBuster mode, you don't see bleaching so much, but you see disassembly before bleaching.
Yep.
So that to me, I mean, Luke's on the call as well, but to me, it speaks that you only need a minute amount of dyes to generate the ROS, right? You don't need all of them to make the damage. Or is that a right interpretation? Or how do you see this?
We haven't. So one really good thing to do would be to titrate different concentrations to look at the percent occupancy of the actual halo tags to figure that out, which we haven't done. I do think you need to have extensive coverage of the intermediate filament network. So I don't know if the amount of dye is more important or the expression level of your vimentin halo tag is more important in this, but it's certainly something that we should test. But I think it also speaks to just how bright and stable these dyes are, that you can use them at incredibly low laser powers and still form images. You really have no business using them at these higher laser powers, even if they are still photostable at those levels. But Luke could unquestionably answer the question better. If another time, I don't have to put on the spot here. Yes.
Do you think you might want to come? Can you speak into the green thing thingy?
Do you think the fluorescent protein like Minnesota will also produce similar effects? If so, then you can couple both to do like dual color manipulations or other things.
Yeah, so I haven't tested Minisaga. I would suspect, based on the performance of Supernova 2, that these genetically encoded photosensitizer strategies may not be as efficient at breaking them apart. But what we actually were able to do is Luke designed, or his group designed, biotinylated JF dyes. So JF 635 biotin was one of the dyes that we used. We were able to disassemble that and then 24 hours later mash up the cells and pull down. Actually, we're still in the process of pulling down. I'm not a great biochemist, but trying to pull it down to isolate to do mass spec. So kind of using the versatility of the halo tag system, there are a lot of really cool options that are available to you. You could throw a sensor on there. You could use one of those inducible dimerization systems in addition to the fragmentation. So having that be the framework on which all of this is structured opens up a lot of exciting possibilities, but it's worth looking into.
If no one else has another question, then I have one. I think you said it in a half sentence. So this is irreversible. So after the burst, would this network build up again at one point?
It doesn't. We actually did a pulse chase assay 24 hours later where we labeled all of the newly translated vimentin after the disassembly, and it didn't form into filaments. They formed these aggregates that eventually accumulated in the perinuclear region and turned over, which is distinct actually from some of the established oxidative damage phenotypes, which are thought to be irreversible, or thought to be reversible rather. One other critical difference is all of that is thought to be carried out by Vimentin's single cysteine that it has. It's thought that you modify that cysteine and that drives disassembly. This process is completely independent of that. We mutated out the cysteine and it still disassembles it. Rather, it appears like the cleanest signature that we're seeing is oxidation of methionines on the Vimentin upon irradiation, driving the irreversible destabilization. But we need to do more work to figure out exactly how it's happening. And last thing I'll leave you with, here are all the abandoned names for the tool and the designs. Vim Vanish, I just really love that logo, but it ended up being a terrible name. But I guess with that, just thanks everybody for listening.
There's a question from Tim, maybe.
Just real quick, can you hear me? So
Oh, I did. Yep.
any speculation on why the snap tag didn't work? Is this a distance thing or a die orientation thing, or any thoughts?
It could be a distance thing. I was loath to show this slide because the real vimentin structure people bristle at it. But when we did AlphaFold of vimentin with the halo tag on it, not only was the halo tag bent back onto the rod domain of vimentin, but it was positioned in such a way that the closest point of contact was actually where the ligand was bound. So it may just be that halo tag is a really favorable conformation, and snap tag may simply not adopt that same conformation. Surely in the AlphaFold that we had, it looked like it didn't. Also, the dyes may not be binding as efficiently, so you're having less dye and less efficient FilaBuster.